Dear participants, welcome you again in this course Systematics of Angiosperm. Earlier we have studied the evidences of morphology and anatomy, taxonomy, and today we will going to study about the evidences of palynology and embryology in plant taxonomy. So, as we have seen earlier that the many evidences of anatomy has helped in identifying different groups of plants. So, today we will going to study the evidences of palynology and embryology. So, today's the content is the palynology that help in identifying the plant taxonomy that had a different relation of the. Then the next one is the embryology in relation to taxonomy. So, starting from the first topic, that is the palynology in relation to taxonomy. As we have seen, the palynology has a different evidences. Like palynology, we have already seen that the palynology is the study of pollen and the spores. So, it deals with the study of pollen grains, either modern or fossil types, and the spores generally focusing on the wall structure rather than on the internal or living feature of in its application. So, the taxonomic characters of pollen grains include wall structure, polarity, symmetry, shape and grain size. And in angiosperm, pollen grains are of two types, that is the monocolpate and tricolpate. And monocolpate pollen grains are boat shaped, have one long germinal furrow and one germinal aperture, as you can see in this particular image. These are the monocolpate. This is the characteristic of primitive dicotyledons such as the Pipraceae and Chloranthaceae, and majority of the monocotyledons like and cycads and seed ferns. Then, according to some palynologists, the first flowering plant had monocolpate types of a pollen grain. Then the next type is the tricolpate grains or globose, symmetrical, have three germinal apertures and are characterized by of advanced dicotyledony. As you can see, these are the tricolpate type, and this one is the equatorial view and this one is the polar view. You can see here, this is the three colpus, these are the equator, these are the pole, this is the polar axis, this is for the further equatorial view. This one is the tricolpate type of a pollen grain. So, and if we see the outer special covering of pollen is called as the sporoderm, which provide another important character for the consideration. Sporoderm or pollen wall is made up of biologically very resistant substance called as the sporopollenin, and pollen wall comprises of a two layer. The outer is termed as the ectexine or also called as sexine, and the inner is the indexine or nexine. And sexine presents various structure and sculptures element which help in the identification and classification of the particular plants, and nexine does not represent such structure and sculptures, but it is thickness or its irregularities help in the various ways to identify different groups of plants. So, if we see, you can see these are the different types of sculpture that is a part of the sexine. So, basic evidence characters are pollen unit type, pollen grains polarity, pollen grain shape, pollen grain symmetry, pollen grain nuclear state, pollen wall architecture, exine stratification, exine structure, exine sculptures you can see in this particular image. Then aperture type, number of aperture, aperture position, aperture shape, aperture structure. These are the different characteristics that use in the palynology for the evidences as a use in case of a taxonomy. So, if we see the some groups, like in case of a Magnoliidae, the pollen grain is nucleate type, and in Caryophyllaceae the pollen is trinucleate, and in Ericaceae the pollen is tetrade. Then in case of a Asclepiadaceae, pollen grain remain in case of a pollinia, that means all the pollen grains are there jointed together and they form of a pollinia. Then Taxacarum, the pollen wall is echinate, and in Quercus the pollen wall is scarbate. So, if we see that there are different types of the pollen characters, and if we analyze them, that termed as the NPC system. The NPC analysis can be applied in systematic investigation in various taxonomic groups like subfamily, genera, or species, like the pollen classification based on the number, position and character of aperture called as NPC system. So, under this system, the usage of term stream have been recommended in place of aperture for the purpose of preparing key for the classification of pollen grains. and a treme is represented by N0 for pollen gland without aperture, and depending upon the number of aperture they may be termed as the monotreme, ditreme, tritreme, tetratreme, as you can see N1, N2 or N3 respectively. And now classified the plant kingdom depending upon their characters like Amorphophytas or Primophoraoreti, which includes thallophyte with no aperture markings. Then Trimophorsophyta, that is also called as the Trimophosphorophyta, which include the archegoniate with monolete or trilete or elite spores. And then is the next third type is the Polymorphous sporophyta, that is the polymorphosphorophyta, which include the angiosperm with diverse aperture type on the basis of the above morphological situations. So this analysis has thrown light on the phylogeny and evolution of the primitive angiosperms. Then if we see the Isonopallinus and Eupallinus taxa, there are Isonopallinus, that is the constant range of Unipallinus, and Euripallinus, that has the drivers of broad range, or Multipallinus with the two broad categories of angiosperm taxa based on the range of they have different types of pollen. So Isonopallinus taxa are those which bear constant pollen type like crucifery, while in others called Euripallinus if there are broad range of pollens or different pollen types varying in size, shape, aperture, exenes stratification like composite that are now termed as the stressing. So if they have a simple constant type of pollen, they are termed as the crucifery. If they have a variety, then they are termed as the Europallinus. Then palynological studies have helped in the research of different taxa like the separation of Peonaceae from the Ranunculaceae, Nelumbaceae from the Nymphaceae, Femiraceae from the Pipiviraceae, then Bombaceae from the Malvaceae and Cuscutaceae from the Convolvulaceae. So according to Bailey and Nast, there are the families of Trichotyledon in which the pollen is very considerable taxonomic significance, not only in the differentiation of the families and tribe but also at the level of genera and species. So we can see the palynology is very important for the identifying the different groups of the plant, not only in the family level but also at the genera or species level. So the next evidence is the embryology that also help identify the many families of the in angiosperm, especially in the taxonomy. So embryology is the branch of the science that deals with the study of sporogenesis, gametogenesis, fertilization, and the formation of zygote and embryo, development of endosperm, finally in the form of a seed and its protective covering, that is the seed coats. So the recognition of the value of embryology and taxonomy as a taxonomic tool for solving taxonomic problem was delayed because of the time and trouble involved in the embryological data collection, because it requires a number of times in collection of these data. The role of embryology as a taxonomic tool in solving taxonomic problem was first brought in the prominence by the German embryologist Snarf in 1931. And although the possibility of utilizing the embryological characters in taxonomy was indicated by some earlier botanists like Hofmeister and Strasburger. And if you see the different features of embryology that used in the taxonomy are the basic evidences are another loci number and arrangement, then anther wall formation and endothecium type, and archaeosporial cell number, aerial presence, embryo sac development type, embryo and embryogeny type, endosperm type, then the integument number and structural type, ovule orientation type and position, teptal type, perisperm presence, nucleolus character, and the hostile function type. These are some features that help in embryology to identify different types of groups and species or genera. So if we starting from the first, that is the anther, the number of arrangement of anther lobe, that either they are monothecous or bithecous. Sporangium is either tetra, bi or monosporangiate. Then the teptum tissues, whether glandular, that means secretory, or amoeboid in case of a periplasmodial, or persistent, or deciduous, dimorphic or monomorphic like in case of Ehrlich Terra thompsoni. So there's a different types of ductal tissue. Then the types of endothelium, like the structure and thickness. So according to Fossard, endothelial thickening contained high amount of alpha cellulose. Lignin was found to be absent from the endothelial wall in the case of a Chenopodium rubrum. But Vasil confirmed the slight lignification at the maturity with a small amount of pectin in case of a Pisum and Lens. Then the modes of pollen grain development, organization and pollen wall stratification, number of cells at the time of anthesis, and the number position of germ pores and the NPC number, as we have discussed earlier, are very important in the identification of different groups of the plants. Then Wadehouse has proposed the classification of plants on the basis of the critical studies of pollen grain structure in various groups of taxa, suggesting that the pollen grains related to the families are similar. Then the next important character in the embryology is the ovule. Like development and structure of ovule, placentation, vasculature of embryo. The next feature in the embryology is the ovule. And the development and structure of ovule, placentation, vasculature of integuments, and orientation of ovule has proved important. Like orthotropous ovule has been considered as a evolutionary older. Then according to Davis, 82% of angiosperm families show allotropous ovules, and synanthropous ovule is characteristic feature of the family Cryptaceae. And if we see the form and extension of the nucleus, whether it is a broad and massive as in case of a crassinuleate, or ephemeral in case of a tunicellate, and presence or glandular deposit of nucleus. And the extent of origin of spontaneous tissue in the ovule, whether single celled or several celled, presence or absence of wall types, types of division of nuclear epidermal cells, mode of megasporogenesis, arrangement of megaspores, the position of functional megaspores, and the development of embryo sac, that is the whether monosporic, bisporic, tetrasporic embryo sac and their types. Then the number and distribution of nuclei in embryo sac and presence and absence of endosperm. Entrance of pathways for pollen tube to ovule, that is porogeny, chalezogamy, mesogamy, or temporal relationship with between pollination and the fertilization in addition to formation of seed and seed coat, have provided data for the taxonomic significance. The next important feature in case of embryology is the endosperm. It consisted of a presence and absence of mode of formation, whether nuclear, cellular, or halobial type, the nature of food reserves, persistence or glandular appearance of endosperm at the time of maturity in seed. Then there are some special cases and abnormalities in development such as the apogamy, apospory, diplospory, adventive embryology, polyembryony, and parthenogenesis. So these are some different characters that can be used to identify the different groups of plants. So if we see, the majority of angiosperm generally, as we have seen that they have an anatropous type of ovules and polygonal type of embryo. This one is the most common in the most of the angiosperm. Then another characteristic feature of the flowering plant is the double fertilization and triple fusion and post-fertilization development of a polyploid endosperm, which is support the probable monophyletic origin of angiosperm. Then the further classification of angiosperm into two groups, namely monocotyledon and dicotyledon, is based on the cotyledon present singly or in pairs, which is the characteristics of the embryo. Then certain groups have well marked their characteristics like embryological features like endospermic or non-endospermic seeds and the seeds with very small embryo, etc. that can be easily used in addition of members of particular taxa. Then the delineation of angiosperm in the first instance as has been largely based on the embryological characters. So as we have seen, there are many characters of embryology can be used to identify particular groups. There are some following are some examples of angiospermic orders and families, member of which exhibit uniform embryological features. Like the Caryophyllales, the order Caryophyllales is characterized by bitegmic ovules with the inner integument longer than the outer, with crassulated type of development, and which are campylotropous or ampylotropous type of orientation and trinucleate pollen grains. So as you can see, this whole order has this type of a embryological characteristic, and the embryo is curved, more or less surrounded the food storage tissue, and consists mainly of perisperm with little or no endosperm. Then in the gentianales, it is characterized by the simple and opposite leaves, absence of integumentary teptine and endosperm development is nuclear type, and these characters help into the distinguish from the other sympatless order. And some families like Burgessiaceae, Menithesii and Oleaceae, which had been formerly placed under the gentianales, have now been removed from it on the basis of presence of integumentary teptum and cellular endosperm. Then the next is the orchideals, and this order is characterized very small seed without a little endosperm or dust seeds. Then similarly Onagraceae and Trapezae, while the family Onagraceae is characterized by the presence of peculiar monosporic tetranucleate onagrate type of embryo sac, like except in case of a genus Trapa. Then the genus Trapa having an 8 nucleate embryo sac, which was earlier placed in the family Onagraceae and since have been removed from the family Onagraceae and now assigned to the new family, that is the Trapezae. Then embryological evidences, however, strongly support the Trapa has well developed suspensor hostorium and 8 nucleate embryosome, which was absent in case of the family Onagraceae. That's why Mansi Ram confirmed this is the Trapa bispinosa. Then the next group, that is the Cypressae and Poaceae. In the majority of the angiosperm, the pollen mother cell divides by meiosis and 5 microscopic tetrads, and all of which remain functional and produce 2 or 3 celled pollen grains. But in the family Cypressae, while all the microscopic nuclei are produced after meiosis, 3 of them are cut off on one side and one of the pollen grain, and only one of the 4 develop to the form a generative cell and then male gametes. So, earlier systematic considered the Cypressae and Poaceae family to be closely associated. However, morphological and anatomic features and the structures of spikelets reveal that two families are quite distinct. Like the Cypressae, Poaceae also delimited by the embryological evidences, like the microspheres in case of a simultaneous type in Cypressae, and out of 3 the tetrad degenerate and only 1 remain functional. On the other hand, in the Poaceae, microspheres is the of successive type and all the spores in the tetrad remains functional. This is the one of the main distincts on the basis of embryology in the Cypressae and Poaceae. Then partial cell is present in case of a archisporium of Cypressae while lacking in case of a Poaceae. The parietal cell is present in the case of a archisporium of Cypressae while lacking in case of a Poaceae. Then antipodals are epiphemeral in case of Cypressae while they are formed complex or become synthetic in case of a Poaceae family. Then embryogeny is of onagrate type in case of a Juncus variation in Cypressae and it is a variable in case of a Poaceae. Thus, embryological evidences reveal a close relationship of Cypressae with Juncaceae. Then if we see the next family like the Garryaceae and Ementiferae. The disputed position of Garryaceae and Ementiferae has also been resolved by the use of embryological evidences, like several taxonomists like Engler and Grigg and Engler and Deals have referred them to Ementiferae, while Bentham Hooker and Wangrin included them in high up in the Polypetalous families like near coronary. However, the later placement is more natural, which has been confirmed by the morphological and embryological studies of Hallock. Then the comparative morphological and physiological studies of family also support the view of their inclusion in the corneals. So then similarly Liliaceae and Emeraldaceae, as we see the family Liliaceae has also been resolved based on the morphological evidences, like Liliaceae and Emeraldaceae family are considered as polyphyletic and certain taxa of these families are closely related. Based on embryological data, the certain taxa have been resolved under the family Liliaceae, Emeraldaceae and Agaveaceae. Because we will see later that the Liliaceae and Emeraldaceae share many common characteristics. So, this embryological is very important in the separation of the different genus in these two groups. Then, other systematic classes are also resolved by the embryological evidences. They are like Peonia was previously kept under the Ranunculaceae, but based on the embryogeny and font size pollen characters, this genus has been shifted from the Ranunculaceae to family Peonaceae. Then Butamus is unique genus having monosporic 8 nucleate embryo sac, while Butamopsis and Limnocaris and Hydrocaris have bisporic 5 nucleate embryo sac. Thus, Butamus retains under Butamaceae, while all other taxa resolved under the Alismastaceae. So, you can see there are some taxonomic division in these particular groups. Then the separation of Hippocratia from the Calistraceae into a separate family, Hippocratiaceae, and placing of genus Sphenoclea under the Campanulaceae is based on the embryological studies. Then the splitting of Gentianaceae into Meninthaceae and Gentianaceae, Malvaceae into the Bombaceae, and Malvaceae is also based on the embryology. And the embryology of Limna, Wolffia and Erythema confirm older views of an intimate relationship of the duckweed with the aroids. Then the disputed systematic position of the genus Parnassia has been resolved by the embryological evidences. Like formerly, Parnassia has been treated as a member of the family Saxifragaceae, but dissimilarity of its embryology with the other genera of Saxifragaceae suggested that it should be removed from the Saxifragaceae and resolved under the family Parnassae. That's all for today. These are the some evidences, as we have learned that these are the palynological and embryological evidences. The next we will learn about the cytological and phytochemical evidences that support the taxonomy. So that's all for today. Thank you all of you for joining this session and bearing the patience. That's all for today.